All right, let's dive right into the personal story behind. And this is no exaggeration, one of the greatest scientific discoveries of the entire 20th century. And the source for this whole explainer, James Watson's own book, kicks off with this idea. You see, he frames the discovery of DNA not as some clean, straightforward, logical process, but as something, well, messy, a deeply human story. And believe me, it is a story. It's about ambition. It's about rivalry. It's a high-stakes race to solve the secret of what he calls the most golden of all molecules. We're going to see how this whole thing was defined just as much by clashing personalities as it was by, the actual science. Okay, so our story really kicks off back in 1951. And it starts with a young American biologist, a guy named James Watson. Now, Watson, and he'd be the first to tell you this, was completely and utterly bored by chemistry. But then, at a scientific meeting in Naples, his inspiration just, bam, came out of nowhere. It happened during a talk by a pretty quiet physicist from London named Maurice Wilkins. So Wilkins flashes this X-ray diffraction picture of DNA up on the screen. Now, what is that? It's basically a shadow of the molecule's structure. And for most people, it was probably just a blurry pattern. But for Watson, no. He saw something else entirely. He saw a clear path, a roadmap to unlocking the very secret of the gene itself. He was suddenly hooked. Yeah, that one single image completely changed everything for him. It set him on this brand new course, leading him away from all his old work and straight to the famous Cavendish Laboratory at Cambridge University. So when he gets to Cambridge, Watson doesn't just find a new lab. He finds something way more important, a crucial ally, someone who shared his obsession with DNA. And that ally was Francis Crick. Watson's first impression? I have never seen Francis Crick in a modest mood. I mean, that says it all, right? Crick was brilliant. He was loud. And at 35, he was a mostly unknown physicist who apparently talked faster and laughed louder than anyone else in the entire lab. The thing is, despite their totally different backgrounds, they just clicked almost instantly. They were both absolutely convinced that DNA was the secret of life, and they had this idea that they could solve it by building physical models. We're talking like playing with giant tinker toys, right? It's what the world's greatest chemist, a guy named Linus Pauling, would do, only they were going to do it faster. But it wasn't going to be that easy, not by a long shot. They had two huge obstacles standing in their way. One was an entire ocean away, and the other, just a 2-hour train ride from Cambridge. So their first rival, this was the big one. The legend himself, Linus Pauling, over at Caltech in California. This guy was a constant, looming threat. They knew he was also sniffing around the DNA problem, and they were terrified he'd get there first. Okay, so that's the rival. The second roadblock was closer to home, and honestly, way more immediate. It was the, let's just say, completely dysfunctional team at King's College in London. Now, this is key because that's where the crucial X-ray data was being produced. We're talking the best in the world. But a deep personal conflict between Maurice Wilkins and his colleague, Rosalind Franklin, meant that collaboration had just ground to a halt. The data was basically locked up. And Watson's description of Rosalind Franklin, who he called Rosie, is, let's be real here, sharp and deeply unfair. But it really highlights the personal tension that was going on. This is the kind of stuff that made getting access to that vital data pretty much impossible. So after Watson actually manages to attend one of Franklin's seminars, he and Crick just rush back to Cambridge. They are absolutely convinced they've heard enough. It's time to build their first model. Well, to put it mildly, their model was built on a series of fundamentally flawed assumptions. Okay, so first, they put the backbone on the inside. Wrong. It had 3 chains. Wrong again. And it was held together by magnesium. This was partly because Watson had completely misremembered a key detail from Franklin's talk, something about how much water was in the DNA molecule, a tiny detail with huge consequences. But they didn't know that yet. So, bursting with confidence, they call the entire King's College team up to Cambridge to witness their triumph. And, well, it was a complete disaster. Their excitement turned into pure humiliation real fast. Rosalind Franklin, she takes one look at this thing and immediately sees the fatal flaws. She very curtly points out that their model just totally failed to account for a massive amount of water that she knew from her own very careful measurements had to be there. In Watson's own words, their model was soft. Basically, it was a bust. The fallout from this was swift. The head of their lab, Sir Lawrence Bragg, was absolutely furious about the public embarrassment. He officially banned Crick and Watson from doing any more work on DNA. This was their absolute lowest point. They were officially, completely out of the race. But even after being officially sidelined, a critical piece of the puzzle starts to emerge, and it comes from a completely unexpected source. So there was this clue, and it was kind of hiding in plain sight. For years, a biochemist named Erwin Chargaff had been publishing these strange but consistent patterns in DNA chemistry. The thing is, nobody, not even the brilliant Francis Crick, had really understood what they meant. The data itself was actually pretty simple, but it was absolutely profound. Here's what it was. In any sample of DNA, the amount of the base adenine, which we call A, was always equal to the amount of thymine, or T. And the amount of guanine, G, always equaled the amount of cytosine, C. Always. A equals T, G equals C. So after a chance conversation with another chemist, the penny finally drops for Francis Crick. He realizes, wait a minute, this isn't just some weird coincidence. This has to be a structural rule. It means A must pair with T, and G must pair with C. This was the missing link, the fundamental rule that would govern the entire structure. So this pairing rule was absolutely the key. But to really get how it all fit into the bigger picture, you have to remember the 5 key people in this whole scientific drama. You've got Wilkins, Franklin, Pauling, and of course Crick and Watson. Their whole complex dance of collaboration and competition was about to reach its climax. So, with this crucial new data in hand, and a rule for how the bases had to pair up, could the duo finally unlock the secret of life? They had this critical rule in their back pocket, and it gave them a second chance. But how they got that chance, and what they did with it, well, that's a story for our next explainer.